Namaskar, today we will study about class 12th, chapter number 11, Biotechnology Principles and Process. To understand principles of biotechnology, we need to understand that what are the techniques involved in biotechnology. Among various techniques, there are two main techniques which enabled birth of modern biotechnology, and that is genetic engineering and chemical engineering. So there are two core techniques of modern biotechnology. First one is genetic engineering. In genetic engineering, we change chemistry of genetic material and deal with the introduction of genetic material into organism which ultimately leads to change its phenotype. Here we mean that we change DNA and RNA in laboratory to get desirable features in organism. We all know that genetic material is DNA or RNA and they are made up of nucleotides, each containing a 5 carbon sugar backbone, a phosphate group, and a nitrogen base. By saying change in chemistry of genetic material, we mean change in bonding of these elements and introducing that change genetic material into organism which ultimately express new phenotype. For example, we do not get vitamin A from normal rice plant. We can insert a gene in normal rice plant to get vitamin A. Rice is naturally low in the pigment beta carotene. Our body uses beta carotene to make vitamin A. Golden rice plant contain beta carotene, which is the reason for its golden color. So here is one daffodil plant which has some desirable genes for enzymes. And here we have bacteria, Arvenia urodovera. Both of these organisms have some desirable genes. And this is our normal rice plant, which lacks beta carotene pigments. Beta carotene pigments is a precursor of vitamin A. If we take our desirable genes from daffodil plants and Erwinia urodivora bacteria, which are responsible for the enzymes necessary for beta carotene production in rice plants, and insert these genes into normal rice plant with the help of plasmid and agrobacteria, we will get new genetically modified rice plant, which we call golden rice plant. Now this golden rice plant have genes for required enzymes which are further necessary for the production of beta carotene. Second core technique is chemical engineering. In chemical engineering, we deal with sterilized, microbial, contamination-free ambience to enable growth of only desired microbe cell in large quantities, to manufacture biotechnological products like antibiotics, vaccine, enzymes, etc. Chemical engineering deals with the integrated knowledge of chemistry, physics, mathematics, and nowadays many more disciplines. So in genetic engineering, we deal with atoms and molecules of nucleotides, which are building blocks of genetic material of any organism. We develop an organism with new features. However, in chemical engineering, we create a sterilized environment. Here we use problem-solving skills, mathematical ability, investigative skills. Here we use that developed organisms and upscale them and make them more efficient for larger production of our desirable products. Thank you.